So our next speaker is Professor Hong Liao. Where is Lena? There she is. Professor Liao, somebody who I've collaborated with for a number of years. She's a world leader in the area of root biology and breeding for, one of the very first to breed for improved root traits, root function, she started working on root architecture a few years later. My lab followed. Now she's working on the root microbiome, and we followed. So whatever she works on, we work on next, because I know to follow the really good scientists. So Hong Liao, her program, she's director of the Root Biology Institute at Fujian Agriculture and Forestry University, and it's a powerful, amazing institute, and they do work all the way from the gene to very serious field work, particularly intercropping and effects of that. She's an expert mineral nutritionist. So she was one of the very first to show that root architecture is an important genetic trait for phosphorus efficiency, tolerance to low P in terms of phosphorus acquisition, and then was probably the very first group to actually breed, generate improved cultivars, breeding for improved root traits, and has released a number of cultivars that are adapted to those low P acid soils in South China. So without further ado, I welcome Hong Liao to come up to give her talk.
Good afternoon. Thank you, Liao, for your nice introduction. You forgot to introduce. I follow you to work on alumina, so we follow each other. So yes. Thank you very much for inviting me here. So today I'm going to talk about, actually this, actually the title, Bill, you gave me the title. So I just think the title is very good. So we engineer crop nutrient efficiency through root modifications for sustainable agriculture. Okay, so they didn't change here. Yeah, what happened? So as you can see, as the plant nutritionist, I always use these slides as the first because we got to know there are at least 17 essential nutrients for plant growth, but only 40 of them are mineral nutrients come from soil. If the soil did not apply enough, so we got to, depends on fertilizers. But however, there are too many problems for over-fertilization, especially in the developing countries like in China. So they always cause over-fertilization, always cause a lot of severe environmental problems. Yeah, this phrase. Okay. So especially nitrogen and phosphorus, they always cause like eutrophication. Another side is over-fertilization, always make the soil degradation. So this is a very serious problem. So what can we do? There is a group in China, we got to increase the nutrient efficiency, but it's still not the same. Okay, I'm sorry, I need to move here. I need to see my slides. Yeah. Oh, thank you. So this one still not working, yeah. So increase nutrient efficiency, reduce chemical fertilization, and achieve high yield. So what can we do? We got to develop smart crops. So what that means, smart crops? So if you want to have the smart crops, at least you got to have smart roots, because over 90% of plants, of nutrients, they take up by plant roots. So you want to develop smart crops, smart roots are the first. So today I'm going to talk about something about soybean. So as Liao introduced, we are the lab, we focus on legume nutrient efficiency. So for soybean first, we think about soybean can do nitrogen fixation. So we see besides nitrogen, phosphorus should be the #1 limiting factor for soybean production. So why I look at the phosphorus? So if you want to develop, the smart roots for the smart plants, smart soybean, we got to smart roots for phosphorus. But if you look at the phosphorus situation in the soil, it's less than 1% of phosphorus, which is soluble, could be directly taken by plants. So this is why when you we were working in the field screening, and you cannot repeat in the greenhouse, especially in the hydroponics, because in the hydroponics, you just only work on less than 1% of phosphorus from the soil. So we often go to the field. But for the field, this phosphorus, over 90% of phosphorus is fixed by soil. In this case, okay, let's see, need to come back. So you need to rely on to increase the root activity, like increase X-rays or to activate the plant's interaction with microorganisms. But no matter you increase the root activity or you increase the phosphorus transporter, but you still need to rely on root architecture because the phosphorus distribution in the soil is very uneven. So we need more roots just distribute where they have phosphorus. So in order to work on this, many years, maybe more than 20 years ago, so we get this established as so-called applied co-correction for soybean, and we have done a lot of field screening almost across the whole country of China, so from very north to very south. And we also do this screening in acidic soils, also in alkali soils, just work on how we can get smart roots for phosphorus. Okay, so here is the what results. So in this case, in the field, we work on the semi-quantification for root architecture. If the roots grew like this, we call the shallow root architecture. This one is the deep root architecture. This is the one in between. So look at the results here. This is the results from several years of field trials. And you can see the genotypes, if they have shallow root architecture, they always grow better at low phosphorus and high phosphorus conditions. So this is tells us the root architecture trees are significantly correlated to phosphorus efficiency in soybean co-collection. Okay. So at that time, we also work on nitrogen efficiency, and we do the correlation analysis. You can see the phosphorus efficiency is significantly correlated to nitrogen efficiency. So the two just coordinate together. So what happened here? So when we check about the modulation, and we found the nodule number was increased by phosphorus addition in over 80% of 500 soybean genotypes. So that tells us. So this tells us phosphorus really promotes nodulation. Oh, okay. So we check out the relationship between phosphorus architecture and the nodulation, and we found most of the genotypes with shallow root architecture, and they have more nodules. Seems their correlation is very significant. So we think about the nodulation, of course, is good for nitrogen fixation. Root architecture, as I said before, is very good for phosphorus. Is there something between? Okay, so the relationship between nodulation and the root architecture. So we develop several genetic populations. So here is one of the example, and we said this is the shallow parents, this is the deep parents, and we found the shallow root parents had better nodulation. Okay, so and we check the reals recombinant inbreed lines, and we found the shallow root genotypes of reals have higher biomass and better nodulation. And then we also check about the root architecture, and we found the most root architecture of most reals become shallower. So this tells us here we have done the same thing, use the semi-quantification method. So we found it's like if you look at the reels, you find that this is the without inoculation. This is with inoculation, rhizobium inoculation. You can see the root architecture go to shallow. So this tells us there should be some like synergistic interactions between root architecture and biological nitrogen fixation. Is that true? So we work on the QTL analysis, and we identify 3 QTL clusters for root architecture, which linked together with the BNF trace. BNF trace is black. I don't know where is the black. Okay, here. So, and also along with the previously identified QTLs for yield. So this tells us it's true. Root architecture, BNF, and yield, they are really linked together. Okay, so we also analyzed the phosphorus efficiency. Not surprise, we identify 2 QTLs for phosphorus efficiency. They also collocated together with root architecture and BNF. From the one of the QTL, we found a gene we previously published before. We found that gene is critically regulate soybean root architecture in response to phosphorus, which is GMEXPB2. Okay, here, good. This GMEXPB2, it is a cell wall beta expansion gene. Now this gene, you can see the expression of this gene is enhanced by phosphorus starvation, and also this gene is expressed only in the root tip, also in the root primordial. So overexpress this gene, just only 2 days, you will see they really change the root elongation and also the performance of root hairs. And this is the overexpressed gene. Even in the very seedling stage, they can increase the plant growth. So this tells us this is a very useful gene to increase soybean phosphorus efficiency. Very surprisingly, we checked the gene expression in nodules. We found that besides the roots, this gene is highly expressed in the nodule, especially in the young nodule. And if you look at the GAS study, you will see, yes, this gene is highly expressed in the young nodules, especially in the vascular tissue. Overexpress this gene, again, the change in root architecture and also the root hair performance. So we use the GFP labeling rhizobial strains to infect this gene. You can see they overexpress this gene and make the rhizobium very easy to infect the root hairs. So in result, they overexpress this gene, they can increase not only nodule number, also the nodule size. So we evaluate in the field and found they can increase both phosphorus efficiency and nitrogen efficiency. So this is another side to prove why we identify 3 QTLs, and one of these really collocated in one of the QTL. Another gene I want to introduce here is the homologue of GMEXPB2, which expression is collocated with one of the BNF traits. Okay, so the 2 parents, they really differed in this gene expression and modulation. So you look at there are 5 sleep different in the promoter region, and this one parent is almost have double nodule numbers than the other one. Then another one, if you look at the nodule development, you can see the infection cell, they just develop much better than another one. Okay, so altering this gene expression really affects the modulation. So overexpress this gene, they increase the nodule number. Unlike this gene, they decrease nodule number, and also affect the nodule development. Remember, GMEXPB2 and this GMINS1, they are homologs. So we do the double RNI, and we found, so if you double RNI these two genes, they really have just almost no nodule formed in the soybean. So so in conclusion, these two genes could control the soybean nodulation, including initiation and development. So another story I want to introduce today is these two parents, they are not only differ in the nodulation and also differ in the response to the inoculation. Okay, so we ask the question. So before we say two genes control the nodulation without like exogenous inoculation, but once we inoculate with exogenous rhizobium, so they change the architecture. So what happened in the microbials in the rhizosphere? I'll come back to look at these three QTLs. We get 3 QTLs to control the BNF in soybean. And we select 6 recombinant inbreed lines, 3 of them without carrying any of this QTL, and 3 of them, this one is carrying all this QTL, and these 2 just carry two of the QTL. And you can see, so without carrying any of the BNF QTL, they're almost low nodule. So they have very small nodule number, and with carry 2 or 3 of this QTL, they have a lot of nodules, especially this one, they have 3 of them. And we work on the microbiome in the rhizosphere. So you can see the soybeans recruited specific rhizobiums and host a distinctive rhizobacteria community from the book soil. So you see, here is the book soil, this is the soybean, and they have a lot of difference. If you look at the QTL, the genotypic difference, the soybean genotype, particularly the 3 QTLs, now here the number represent the different QTLs. The 3 QTLs, they greatly influence the structure and the composition of rhizobacteria community. So you will see, so they are clearly separate from each other. And we work on the bacterial network. So we see these 3 QTLs, they changed the composition of the network, and also they changed biomarkers of the network. So this tells us the genetic trace and the exogenous rhizobia synergistically regulate soybean rhizobacteria communities. So what this for? Once we get all those QTLs, we work on the mark-based assisted selection breeding for new materials with superior phosphorus efficiency and better nitrogen fixation for sustainable agriculture. So here I wanted to tell you something about what we have done for the sustainable agriculture. So we use those, we developed some phosphorus efficient soybean varieties, and we grew them in the T soybean intercropping system. So here is the T soybean intercropping system. You see the light green are soybean, the dark green are tea plants. So in the tea plantation, the soil are very acidic. The pH is almost, both of them lower than 4.5, the lack of NPK and toxic in alumina and manganese. And we grew this tea soybean intercropping. The tea soybean, they both grew very well. You see the soybean are very happy in the tea plantation. Even in these very hilly places, the soybean grew very well. And we look at the nodulation. They have very good nodulation, very well. Oh, okay. So after intercropping with the nutrient efficient soybean varieties, you can see they can increase the tea yield. So for the young tea trees, they almost double the yield. For the old tea trees, they can increase about 50%, 40%. More interesting, so when intercropping with the soybean with the tea plants, so you can see the reduced insects and diseases. So I have the video, very beautiful, but I'm sorry, you cannot see that. So the soybean just attract all the insects. So the farmers tell me, I don't worry about that, but I'm not really care about how to increase the yield. I care about you don't need to use the pesticides. So this is a more environmental friendly system. Okay, we also checked soil fertility changes. So intercropping with the soybean plants, you can see the increase before the pH is very low, so they increase the pH a little bit, and also increase the available nitrogen content. It's not a surprise. So they can, this is maybe partially explain why they can increase the tea yield. And more importantly, this soybean tea intercropping significantly improved the eco-environment of the tea plantation. So this is just like they make the tea plantation is a very happy places for all those birds and good insects. So we dig out the roots and we found the soybean roots and the tea tree roots, they're just very happy they attract each other, very happy to stay together. And the soybean also, you can have very good soybean yield. And we also work on the microbial analysis in the tea plantation, the soil microbial analysis. And you see, this is the 1 year intercropping soil. This is without intercropping. This is 2 year intercropping soil. So you can see they really change the soil microbial community in the soil by the intercropping with soybean. So this is a very good system. So now we are just like extend this system in Fuji agriculture. So finally, I got to thank Liao again for inviting me here, and also I would like to thank all my RBC members and my collaborators. Thank you very much for your listening.